Hi folks, welcome to my channel, The Average Dermadoc. Let's begin the show. Biologicals or biological products are produced in a living system. Bio means living system. These are the products produced from the living systems like microorganisms, plants or animal cells, which includes vaccines, therapeutic proteins, monoclonal antibodies, even blood and blood products also comes under this biologicals. This production is limited, hence they are extremely costly. To produce biologicals, we need a living system. It requires a huge setup. So, very few companies are producing biologicals. Hence, they are extremely costly. Only less than 5% of the population are being utilizing these biologicals. Mostly, they are also from state sponsorships. That means in my state government is giving rituximab at free of cost to patients. Those 5% population also mostly falls under this group. First biological approved by FDA is INI-CET, which is used for chronic plaque psoriasis. Coming to classification of biologics, they are classified into three types. One is based on molecular structure. One is based on their mechanism of action. Another one is based on their indications, where they are being used. Clear? They are classified into three types: based on its structure, based on its mechanism of action, based on its indication, where and all they are used. Coming to one by one, based on its molecular structure, whether they are monoclonal antibodies, fusion proteins, recombinant human cytokines. Cytokines are nothing but interleukins, interferons, growth factors like epidermal growth factor, fibroblast growth factors, insulin-like growth factors. This classification is based on their molecular structure. Coming to monoclonal antibodies. Name itself telling us that these are produced by a single clone of cells. Which cell will produce antibodies? B cells. B cells are mortal, that means they will die, but B cells will be combined with a immortal cell using genetic engineering, recombinant DNA technologies, and a new hybrid cell will be produced. These cells are called hybridoma cells. We will have the property of a B cell producing antibodies, and we will have the property of a immortal cell. That means it will be producing without dying more and more antibodies. Such cells are called hybridoma cells. No need to remember all these things. Just monoclonal antibodies are produced from a single clone of cells. All of them have MAB as a suffix. That means at the end of their name, MAB will be there. Then we can easily identify it is a monoclonal antibody. Before that suffix, that is prefix, indicates their origin. Based on their origin, these monoclonal antibodies are divided into three classes. Which denotes origin? That is prefix. Prefix is just before suffix. Here you can see in rituximab, MAB indicates it is a monoclonal antibody and it is suffix. Just before suffix, that is prefix. The letters are XI. Study this XI like chi. These are chimeric antibodies formed by fusion of human and non-human species like murine species, mouse. Antibodies will have a constant portion and variable portion, that is antigen binding portion. Constant portion is originating from humans. Variable portion is from mouse. Both combinedly, monoclonal antibody is called a chimeric antibody. Because of this non-human species portion, that is foreign to us. Any foreign body, if it enters our body, it will stimulate our immune system. That's why these chimeric antibodies will have more immunogenic property. Nearly 40% of the monoclonal antibodies portion is from murine species in chimeric antibody. To reduce this non-human part, they have produced humanized antibodies with prefix ZU. Remember, humanized ZU. it will have murine component less than 10% only. Hence, it is less immunogenic. For example, omalizumab. It is used in chronic spontaneous urticaria. It is an antibody against IgE antibody. Next, prefix U, fully human antibody. Example, secukinumab, adalimumab, just U, fully human antibody. Fusion proteins, two or more genes are joined or fused to code a new protein, that is fusion proteins. Example, receptor fusion protein, alifacept, etanercept. Next, recombinant human cytokines and their antagonists. Example, interleukin-1 receptor antagonist anakinra, used in rheumatoid arthritis. Next classification, second classification, is based on mechanism of action. Whether they are acting on cell surface receptors, co-stimulatory molecules, cytokines, or cytokine receptor antagonists. We will see one by one. Biologics acting on cell surface receptors, that is anti-CD20 antibody. This CD20 is a receptor on B cell. Example, rituximab. From now onwards, if you see any monoclonal antibody, you have to be able to tell its origin. That is, here you can see XI. I have told you to read this XI as chimeric antibody, used in immunobullous diseases. Next, anti-CD3 antibody. This CD3 is a cell surface receptor on T cell. Muromunab, used in transplant rejection. Here, this antibody is a monoclonal antibody, but it doesn't end with MAB. Before this standardized nomenclature came, this antibody was produced and named so that only this antibody don't have MAB at the end. It is a chimeric antibody which is no longer available now. Next, which acts on co-stimulatory molecules. Anti-CTLA4 antibody. CTLA means cytotoxic T lymphocyte antigen 4. If this antigen is suppressed, cytotoxic T cells will be free to chase and kill melanoma cells. Wherever these melanoma cells are hidden in the body, even if it is a metastatic melanoma, it will go and kill that melanoma cells. That much efficacious, targeted and powerful these biologics are. Example, ipilimumab, which works against melanoma. Mumab melanoma. LFA3 CD2 interaction inhibitor alifacept, used in chronic plaque psoriasis. We have already seen alifacept is the first biological approved for chronic plaque psoriasis. Next, cytokines are cytokine receptor antagonists. TNF alpha inhibitors, adalimumab, infliximab, etanercept, certolizumab, golimumab. We all must have heard all these names. Used in psoriasis, pyoderma gangrenosum, hidradenitis suppurativa, Behçet's disease. Interleukin-17 inhibitors, secukinumab, brodalumab, izekizumab. Interleukin-12-23 inhibitors, ustekinumab, 23 inhibitor, guselkumab, tildrakizumab. All these are used in psoriasis. Clear? TNF alpha inhibitors, interleukin-17, 12-23, 23. All these are used for psoriasis. Next, interleukin-4-13 inhibitors, dupilumab, 5 inhibitor, mepolizumab, 31 inhibitor, nemolizumab. Dupili, mepoli, nemoli. All these are used in atopic dermatitis. Next, last classification, it is based on indication. For example, chronic spontaneous urticaria, omalizumab. Here you can see for atopic dermatitis, all these are used. Psoriasis, all these are used. So you can easily write disease name and its biologicals being used. Coming to biosimilars, these are also biologics only, but they are highly similar to an already approved reference biologic, with no clinically meaningful difference in safety and efficacy. already a biologic is there. Based on that, similar product is produced. That is why it is called as similar, biosimilar. It doesn't have any difference clinically in efficacy wise, safety wise. These biosimilars are more affordable alternatives to these biologics. For example, infliximab is a biologic. A similar product is produced, CTP13SB12, which is clinically equally efficacious and safety. Why is also same with infliximab. That's why they are called as infliximab biosimilars. Adalimumab biosimilars, etanercept biosimilars. These are all examples. How these biosimilars are made more affordable. Why biologicals are so costly? Biosimilars are more affordable because they are produced by many companies. Once a biologic got approved, it will undergo clinical trials and it will be available to normal population with patency. That means a company will take patency of its own biologic at least for a minimum of 20 years. After 20 years, this patency will get expired. After this, many companies will come and decode that original biologic composition, which will be very complex, and so that these companies will be easily decoding that original biologic and produces a new similar product called biosimilar using reverse engineering, bioreactors, etc. It is very difficult to code a product and go all the clinical trials, but it is very easy to decode that product and produce a similar product, not same product but similar product. Here it will undergo extensive analytical comparison whether it is structurally, functionally, pharmacokinetic and pharmacodynamic similarities are there or not with biological. But these biosimilars won't undergo much clinical trials. So no need to go much clinical trials. Already marketing will be done for that original biologic. So many companies are producing. Hence availability will be increased. So that cost will automatically come down. So hence it is more affordable alternative to biologics. Until that patency gets expired, these companies will wait to decode that biologic and produce biosimilar to make it more affordable. Coming to pre-treatment evaluation for any biologic. If you want to start a biologic to anyone, with any biologic you have to do pre-treatment evaluation, which is same. That is, you have to take history of active or treated TB, diabetes, hypertension, cardiovascular diseases and demyelinating diseases like multiple sclerosis, Guillain-Barré syndrome, history of jaundice to find out any hepatitis is there or not. Recent travel. If you travel recently to any place, there you will eat new foods or you will drink new water, there you may get jaundice, or you may get exposed to new people with high risk sexual behavior. Vaccination history, if you have taken live vaccines or not. IV drug abuse history, all these are to find out STDs, whether you have got exposed or you are having any STDs or not, mainly HIV. In HIV, we will be already immunosuppressed. Remember the channel name. Don't forget to like, share and subscribe. Thank you. Hasta la vista. Desvidania.